Hello, everyone. This is Professor Sen, and I'm making my first ever Nanopore data analysis tutorial. It's a newer DNA sequencing technology that some people are calling third generation sequencing, where we get really long reads and we're able to get just about the full length of our 16S gene, which is that gene that's highly conserved in all of the bacteria. So all of the bacteria have it, and it's also variable, meaning that we can use it to identify the different genus or species of bacteria. So this data is our first run, and it's from the Rodale strawberry cover crops trial, where they were comparing plastic row covers, which is the traditional practice, and comparing that with different types of cover crops. So let's take a look. So for this, it requires some processes before the data analysis itself. So first, the soil was sampled from the field, then the DNA was extracted from the soil, and it included all of the DNA from any organism, living or dead, that was present in the soil. The next step was to do PCR, which is part of the library prep process. And the purpose of the PCR was to amplify just the 16S gene, from the bacteria. And then a barcode is attached that allows us to put all of the samples together in the sequencing, so it saves a lot of money. And then we can separate them using the computer, using bioinformatics, by those different barcodes. So the barcode basically identifies each sample so they can be put together and then later demultiplexed. So after the library prep is the sequencing step. That's when we get our raw sequence data, base calling. That step converts the raw sequence data into the A's, T's, C's, and G's that are part of the genetic code. And then there is demultiplexing, which is that step where the sample data is binned based on the different barcodes. So then we get a separate file for each sample. And those files are inputs for the next step. So different input files that you'll be using for this tutorial to get your graphs for your final project and final report include the metadata. So the metadata is basically the background information about the samples. In order to keep track of which samples belong to each treatment group while you're doing your analysis, you should have your metadata table handy for viewing. The best way to look at this is going to be in Excel. So you can go to Excel, open it up, and then when you open file, look for your metadata file. And the version that you have is a text file. The metadata table can also be used as an input in later analyses that aim to compare groups and analyze potential differences in treatment statistically. So it's a required input for some of the downstream analysis. One of the main files that you're going to be able to get graphs from today are the Workflow 16S sequencing report. So the Workflow 16S is an app, or it's a workflow inside of the Epitome app, where the sequences made up of those A's, T's, C's, and G's are classified or assigned to different taxa. So for example, you might have some samples that include the genus Bacillus. So you're going from the sequence, and then it's blasted in a database on NCBI to try and identify what's the most likely taxonomic classification for that bacterium. And it's done over and over again for all of the sequences or reads in each sample. So now that they have been named to specific bacterial genera or families, this means that you can now interpret the results, because this step determines the counts of the sequences from the different types of bacteria that are present in each sample. So the workflow 16S sequencing report provides summary statistics about the data, like the amounts of each bacteria sequence found on a sample by sample basis. So you have to go into the drop-down menu and actually select your sample. And it doesn't allow you to compare them statistically just yet in this program, but the outputs like the taxonomy abundance table can be used for statistical analysis later. I'll give you some suggestions for additional visualizations that you can make. Since the bacteria has been classified by this step, by the workflow 16S, the output also shows you a tree and a sunburst diagram that will allow you to explore the bacteria that were present in your sample by the different taxonomic levels. So similar to what you may have seen in a program like DNA Subway, you have little drop downs where you can select the different taxonomic levels like kingdom, phylum, class, order, family, and genus. And then it will display those graphs based on the different levels or the level you select. So the information that's in the workflow 16S sequencing report can allow you to pose and respond to a few different questions. So first, you have the read summary. And with this information, you can ask the question, how high was the data quality? Also, how long were the sequences that were ascertained by sequencing? The expected length of our bacterial identification gene, or the 16S gene, is around 1,500 base pairs. So we're certainly expecting these amplicons or the PCR products that were created in the library prep to be greater than 1,000 base pairs. Okay, what else can we ask here? Which samples had the most reads? In other words, which samples had the highest count of bacteria? Or you might say, which samples had the most bacteria overall? Not thinking about taxonomy, just like who had the most, period. And then how do the samples compare in terms of their composition at a selected taxonomic level? So we have a graph that you'll be able to see in just a moment for that. And then how many total species were identified in each sample? So let's take a look at this workflow 16S sequencing report. Okay, so as I mentioned, the first part of this report is the read summary. And here you can find out about the quality of the reads. In order to have passing sequences, I believe they have to be greater than 9, so like 10 or above. And most of these sequences have good quality. The example I'm looking at right now is just sample 1 because that's the one that comes up first. Next is the read length. So here we have the x-axis, the read length in kilobases. So that's like 1 would be 1,000 bases. And then we have number of reads, which is like the count of the total reads at that length. And that's on the y-axis. Okay, next we have the base yield above read length. And here we have a curve for that. Okay, so the next thing here is the table that gives the number of total reads. So here we can ask that question, which samples had the most reads? Or you can say, did my sample have high or low number of reads compared to the other samples? In other words, which samples had the highest counts of bacteria? So here you can actually sort these. So if I sort them with the up arrow, it's going to sort ascending. So starting with the smallest values, working up to the highest values. And unfortunately, you can see that samples 4, 7, and 9, or plots 4, 7, and 9, those samples had very few reads, didn't come out too well. Unfortunately, those we would just have to throw away. But any of these, like starting with 8, which has 81, or any of the samples that have above 81, we should be able to get some sort of information from. And then Unfortunately, there were a few samples that failed sequencing on this run. Like I said, this is our very first time. So that's why it'll be useful to have your metadata handy, and you can look and see if, your sample failed. For example, if you had sample 9, then you can find out which samples were from the same treatment, and then choose another sample that had the same treatment as yours. That way you'll be able to move forward with your graphs. You're going to need to get your graphs today. Those are due this week. And then when you actually do your final report, you'll have something to write about. Okay, so here it's it's sorted ascending. But if I put the down arrow here, then it'll be sorted descending. So I'll basically get like the maximum value first, and then it will go down. And I looked at these, I just looked at like the top 7 values, or the 7 maximum values that had the highest number of reads. And I found that out of those 7, 3 of them were from the plastic plots. So I got like a little bit of a sense, which was consistent with our artichokes data, that the plastic treatment tended to have the higher abundance of bacteria in general, not taking into account what kind of bacteria, just like they had the highest number of reads. So you can take a look at this, try to find your sample, see which sample you're going to be working with for the final report, so you can get some graphs and get a sense of whether your amount of reads was high or low compared to the other samples. Okay, so this is our tree, and this tree shows how the samples, or excuse me, how the different taxa or different types of bacteria in the samples are related to one another. So here we have sample 1 selected. I'm going to go ahead and switch it up to sample 20. We do see that there's a high number of unclassified reads, so those weren't identified to a particular bacteria. And then we have everything else. So this one is a little bit simple. It doesn't have as many different species in it, but we have 69, this is, I think, under order, 69 Bacillales, so that's going to include the genus Bacillus. We also have here, if you look at the class of bacteria, we have alpha proteobacteria here, containing both the hyphomicrobiales and sphingomonadales. So let's see if we zoom out a little bit, maybe we can see more. I'm going to try a different rank, which is the taxonomic level, and maybe a different sample too. Let's try, here we go. Sample 13. So we can actually take it down all the way to the genus level, because we have classification up to the genus level. So you can use this tree as one of your outputs. And you can even use this to answer the question, how do the samples compare in terms of their composition at a selected taxonomic level? Okay, and then we also have a sunburst diagram, and the sunburst diagram is interactive. Would you like to lose your computer for the rest of the day? Go out and okay. The next one is the sunburst. And like I mentioned, this one is interactive. So here we have going through the different taxonomic levels. So unclassified, we can't really find out too much about it because those are unclassified, they're unknown. But if we go into bacteria, we can go through Bacillus, Neobacillus, Peribacillus for this particular sample, Cytobacillus, Priestia, Russellomorea, Fredonencohinia, Pseudoneobacillus, Ammonophilus, Brevibacillus, and then we have Thermobacillus, Ureabacillus, which sounds kind of funny, and then Tumibacillus. And then, let's not forget about Caldana robius. And then we have rhabdanaerobium. And after that, we move out of bacillus finally. So there were really a lot of bacillus in these results for many of the samples. And then it goes toward Pseudomonadota, which are going to be the ones that are related to Pseudomonas. So hopefully you get the idea here. And we have, for example, like Sphingomonas. Some of these we saw in our previous results, Novosphingobium. And then you can move out of Pseudomonadota, and it looks like we have Actinomycetota. It's going to include the actinomycetes, like our streptomyces. Here we have a few different ones. And then it goes into Myxocochota. So different things that you can explore. These graphs are helpful for your final project as well. Oh yeah. So here, this one actually compares all of the samples. So it's nice because it puts everything into one. It's not labeled by the treatment though. It's just labeled by the number of the plot. So that's something to keep in mind. Right now it's set to the phylum rank, and we can see that there's some differences here. It's a color-coded chart. The different colors represent the different phyla of bacteria. We have the relative abundance on the y-axis and different samples are on the x-axis. So let's change this. I'm going to try family to see if it's more interesting. Okay. Interesting here, that number 9 that didn't have too many reads, it looks like it was 50% Azospiralaceae, so we can get something. And then we have the rest was unknown. So I think it was only like 2 reads here. And you can see the different families. So Bacilliaceae, you can see that one was the one that had bacillus and all the different types of bacillus. It's the main one out of the ones that were identified. And then all of these smaller blocks here are going to represent the other top 8, because I think this is showing the top 9 most abundant families. Okay, then here we have the abundance table. This is going to be the same as our Excel spreadsheet, so I'll go over that next. And then here we have the alpha diversity. So the one that I want you to look for here is the effective number of species. And this is going to tell you the effective number of species that were identified in your sample. And you can scroll across here and be able to find your sample. The sample is on the top row, the sample number is, and then below that, the column below your sample number is going to help you find the cell that corresponds to the effective number of species on this row here. So that allows you to answer the question, how many total species were identified in each sample? Let's go back to our tutorial and talk about a little bit of what to expect from the Excel pivot table analysis. So remember, it's helpful to have your metadata available just in case you need to look up a sample number and just figure out which sample that represents. It's going to be the same as your plot number, but if you want to look and see which treatment different samples belong to, that will be helpful. In our version today, I've already gone through and the samples have been renamed by the treatment group. So I'll show you an example of that in just a moment. Suggested uses of the abundance table, which I thought were pretty fantastic in the midterm exercise. You can filter the spreadsheet of counts to display the top 3 maximum values for your sample. Those are going to represent the top 3 bacteria that had the highest abundance in your sample. And then something that I saw people do that was a little bit above and beyond and fantastic on the midterm was calculate % of the total bacteria for the top 3 bacteria in the sample. You can also generate some graphs in Excel from your pivot table. So I'll show you what that looks like. You're going to create an Excel pivot table that highlights the taxonomic levels and samples of your choice. And you want to then generate at least 2 graphs that depict your results in the graphs visually. It's optional also to filter the pivot table to the taxa of your choice. So maybe you're interested in the genus Bacillus or the family Bacilliaceae, since we had a lot of those in there. Or maybe you're interested in filtering it down to those top 3 maximum values, or those top 3 most abundant genera or families in your sample. So you can filter just to highlight those. Some of the different graphs that you can generate from Excel, you can create a stacked bar chart. The stacked bar chart is like the one that we had in Workflow 16S. The sequencing report, it's similar to a bar chart, or excuse me, it shows the relative abundance of the different bacterial taxa. This is the stacked bar chart, and you can compare samples or sample treatment groups by selecting all the individual in the groups that you wish to compare. The stacked column chart, I should say, it's similar to the bar chart, but just rotated 90 degrees. So it's going horizontally on the bar chart, and then the column chart is oriented vertically. Sometimes referred to as a frequency histogram. So you might have heard that term before. An area chart is another variation of the same that will allow you to examine the relative frequencies visually. You can also generate a pie chart. This one is the classic. It shows the microbial composition of a single sample. So you can generate one of these just for your sample. Or you can use a donut chart, which is a similar output to the pie chart, but it has a different arrangement that looks kind of cool, and it's colorful. So either of those would be fair game. So let's go ahead and take a look at the abundance table, create our pivot table, and then try to generate some of those graphs. Okay, so here it is. The output abundance table from Workflow 16S has been modified just a little bit. It shows all of those different taxonomic levels that we're interested in: kingdom, phylum, class, order, family, and genus. And then in this instance, I color-coded the similar treatments together. And the samples have been renamed by the treatment. So we have like sorghum and peas 1, sorghum and peas 2, etc. Perennial clover 1, perennial clover 2, perennial clover 3. Plastic 1, plastic 2, and plastic 3. And then we do have 2 buckwheat and pea samples here, but the other one was the one that failed sequencing. So we don't have a fantastic representation of buckwheat and peas, but you can definitely take a look at the data that is available and display in the graph. So let's see what we have. The first thing that we want to do is go up here on the top ribbon, and we're going to go to Insert. And once you have this Insert tab available, we'll go ahead and insert Pivot Table. Pivot Table is here up on the top left. And then we'll just go ahead and take it in a new worksheet as it's suggested. So let's see. Let's try and do this maybe by class. Sounds interesting. And then we can select the samples that we're interested in. So I'm going to select plastic as one of my comparisons, since that's the control of the study. It represents the common practice that growers traditionally use. And then let's go ahead and try perennial clover as a comparison. So I've chosen 3 of those. Let's see, perennial clover had 4 samples that made it through. So we might as well take a look at that. If you had a particular class in this instance that you were interested in, so here in the first column, which is column A, we have the different classes. So for example, there's like Clostridia here. You can filter these down. So if I look at bacilli, I can see that we have the highest count, right? There's like 94 in plastic, 67 in plastic 2. The plastic 3 didn't have too many though. Perennial clover, the bacilli, we have 98, 38, and 19. So you might want to look for the ones that have the higher numbers because they may be more interesting for comparison. And then we can also see the total numbers or counts of bacteria when they're summed up here on the bottom row. So I'm going to go ahead and try to insert a pie chart. And I think that this is going to come up for the first sample here. If I select it, let's see what happens. So here we go. I'm able to get here. This is by class. You can check a different box on the pivot chart fields if you're interested. And then I can also make it larger to try and get it to display all of the different classes. So I'll adjust that a little bit to try and get it to show everything. Okay, now it looks to me like that's showing the whole list. And we can also adjust the options for this. So let me try and go to the little plus, and we can try and get a data call out. So if I go for data labels, then it's going to give me the number of counts. Let's see, what else can we do? We can also get it by percentage. So this is kind of fantastic here. Without really having to think too much about calculating it, I may need to move things around to make this readable though. And then keeping in mind that each of these slices of the pie, regardless of whether or not you use the data call out, is going to be color coded. And then if you check over here on the legend on the right, then you should be able to determine which slice goes with which taxa. In this case, it is under class. So, you can play with this a little bit more, trying to spread these out enough that they're readable. This can give you an idea about the different percentages. And then this one was the first sample that was on the left. I'm interested in seeing if I can get this to work for another sample. So let's try 2D pie chart. It looks like it's still doing plastic 1. I think that if I can get my options back here. For this one, if I can remove plastic 1, it might help. So let me try deleting this sheet and starting over just for a moment. So I'm going to go ahead and go to insert pivot table. And I was interested in class. And then if I just select, let's say plastic 2, if you were in that plot, then I should be able to go ahead and go in and insert the 2D pie chart, and then it can be adjusted similarly. And if you look closely here, it is showing me plastic 2 in this instance. So plastic 2, it doesn't necessarily mean plot 2, but it means it's the second plastic sample that made it through. So if you go and correlate that, find the corresponding sample in your metadata, then that should be helpful. Adding my data labels here. All right, great. So let's try and do one of these others. Well, we maybe can use a stacked column chart. So I'm going in, inserting my pivot table, and then for stacked column chart, I wonder about trying this one on phylum. Of course, you have the ability to look at this also on the workflow 16S, but this allows you to sort of slice it a different way and be able to really look into your data. All right, so I'm going to put in phylum, and then I'll choose the plastic samples, and maybe just for my example, I'll choose something else. Something else other than perennial clover this time. Okay, so we have our plastic samples. And if you look at this, when I choose the higher level classification, so in this case it is the phylum, then there are less rows, right? Because it's less specific, so there are less taxa in that classification. Because it's like a tree, right? It has a lot of branches on the lower levels. But close to the kingdom level, which is bacteria in this case, there are less branches, and then the branches get more and more numerous as you move away from that trunk or that base, which in this case is the kingdom bacteria. Okay, so let's try sorghum and peas. I'm just going to go ahead and take the first four because I think with that one, since we had two different sorghum and peas seeding rates, eventually 6 of them made it through sequencing on this test run. So I have 3 plastic, and then there's 4 of the sorghum and peas. And I want to try to put in a chart for stacked column chart. So here it looks like we have a clustered column. Ooh, here's the stacked column chart. This one's cool looking. All right, I'll go ahead and try and make this a little bit larger. And now in this instance, instead of the different colors representing the different taxa, and remember here we're at the phylum level as it shows on the bottom left, each of the colors represents the different samples. So this is like a totally different way of looking at it. You're not just looking at one sample, now you're able to compare the levels from each. Okay, so we have different colors that represent the different samples. They're color coded. The abundance is on the y-axis, and then the different phyla of bacteria are on the x-axis. And you can play around with the options, selecting different taxonomic levels, like you can select genus or others, and then take a look at the differences between samples between the treatments. Okay, and then I'm sort of tempted now to do the donut chart since I made such a big deal about it. So instead of fighting with that, I'm just going to go ahead and create a new pivot table since it's so easy to do. And let's go ahead and do this one with genus, since in that case, we'll have a lot of different rows and a lot of different counts that can be reflected in the donut chart. And you'll see why it might be kind of more interesting than doing the pie chart. Let's take a look. So we looked at plastic 1 and plastic 2. Let's do something totally different. I'm going to select one of the buckwheat and peas. This looks kind of bad. What about the other one? Okay, they just have a little bit of low counts there. But let's see what we can do with our donut chart. So this is another type of graph that you can potentially include. So here, now we're just looking at one sample. So we're looking at one of the buckwheat and pea samples, and then each one of the colors represents a different genus. And of course, I'm expanding out this list a little bit to try and get everybody represented, but the list is really long. So you could also try this and the family level, which will give you less rows, or phylum, class, order, the other things that we've been talking about. All right, great. So I hope that is helpful, and it should give you the opportunity to learn a little bit more about bacteria, find out from this particular nanopore DNA sequencing data set, what were the top 3 maximum values, what were the percentages of the top 3 bacteria or other interesting bacteria that are in your sample. You can find out about your data quality, the length of your bacterial gene that was sequenced, the amount of sequence that was generated or ascertained, which samples had the most reads or the highest counts of bacteria. How did the samples compare in terms of their composition at different taxonomic levels? And then how many total species of bacteria were identified in each sample?